This is Lecture 5, Neuronal Adaptation and Communication, and it corresponds with Chapter 5 of your textbook. Some reminders and announcements. Homework 5 is due as usual this Sunday at 11 p.m. On Monday the 11th, Exam 1 will post, and it's due the following Sunday at 11 p.m. The exam is timed. You'll have 90 minutes to complete it. It's usually about 40 to 50 questions. Questions are comparable to the homework questions. So you'll have multiple choice, true, false, and some essay questions as well. Short essay questions like the ones you get on your homework. And you can use the textbook, you can use lecture slides, your own notes on the exam. I do ask that you don't work with other students or get help from anybody else on the exam. Your recommended readings for the week, and again, these are not required, but there's a book by Oliver Sacks called Musicophilia. Oliver Sacks is a neurologist who wrote a number of books about case studies and sort of about interesting neurological phenomena, and he writes in a way that's very accessible to the common public. Musicophilia is specifically about case studies and in general normal brain functioning and how music sort of plays this unique role in some neurological cases, and also just how music is perceived in the brain in general. You can also get it on audiobook, and if you haven't used your free trial, if you have an Amazon account and haven't used your free trial of Audible that comes with that, you could get it. You could listen to it for free on audiobook through the free trial of Audible if you're interested. There's also a New York Times article that some of you might find interesting, especially people who are particularly interested in exercise, about neurogenesis and how exercise has been linked to neurogenesis. Okay, getting into the course material, this corresponds to Chapter 5, Section 1, Chemical Messages. So we have two main types of chemical messengers that we're going to discuss, and those are neurotransmitters and hormones. Neurotransmitters are chemicals that signal in the central nervous system and have an excitatory or an inhibitory effect on the target when they're released on a neuron. There are maybe 60 to 100 different neurotransmitters in the central nervous system. It can be kind of difficult to decide what's a neurotransmitter, so scientists are still trying to work out exactly what's a neurotransmitter and what isn't. Then hormones are chemicals that are transported by the circulatory system, and they act on organs, but they also affect behavior and general body physiology. The hypothalamus controls pituitary function. It controls the pituitary gland. The pituitary gland releases hormones into the bloodstream, so the hypothalamus is kind of the one ultimately controlling hormonal release. Then hormones affect target glands and organs in the autonomic and enteric nervous systems. Hormones do travel a lot further than neurotransmitters, but they're also slower acting.
These are just some major neurotransmitters in the body, and we're going to be returning to the specific roles of these, but you've probably heard of things like dopamine, maybe glutamate, norepinephrine, which is noradrenaline, serotonin, things like this. So getting back to the neuron, if you remember, the synapse is this junction between two neurons, between the presynaptic neuron, which is the neuron that's releasing neurotransmitters, and the postsynaptic neuron. A chemical synapse is this junction where messenger molecules or neurotransmitters are released from one neuron and they excite or inhibit the next neuron. Most synapses in the mammalian nervous system are chemical, and when we talk about synapses in general in the human nervous system, we're almost always talking about chemical synapses. So as mentioned before, the presynaptic membrane, which is also the axonal terminal, is where the action potential terminates to release the chemical message. And the postsynaptic membrane is after the synapse. This is also typically a dendritic spine, although it can be other parts of the neuron. is the receiving side of the chemical message. So this is where neurotransmitters get released and excitatory postsynaptic potentials or inhibitory postsynaptic potentials are generated. The synaptic cleft is this space between the two neurons. So there's just a very small amount of space. These two neurons are not actually touching, and so the small gap is called the synaptic cleft. Synaptic vesicles are presynaptic. They're small membrane-bound spheres that contain one or more neurotransmitters. Storage granules are also presynaptic, and they're membranous compartments that hold several vesicles. And then postsynaptic receptors are these sites, these proteins that are embedded in the postsynaptic membrane, and these are the sites to which a neurotransmitter molecule binds. And these are just some pictures of synapses from electron microscopes. So these little things that look like bubbles are vesicles, and these contain neurotransmitters in the presynaptic cell. And these gaps you see between the cells, that's the synaptic cleft. So there is a different type of synapse that is much less common than chemical synapses in mammalian systems, and that's electrical synapses or gap junctions. These are ion channels that form a pore through which ions can flow directly, so there's no cleft or no gap between the cells. This transmits information faster than chemical synapses. It is one way that interneurons fire in synchrony, so fire together by using these really quick electrical synapses. It allows for exchange for substances between neurons and glial cells, but chemical synapses are more common in the mammalian brain because they're more flexible, complex, and they can change with experience. So learning and memory are dependent on chemical synapses. So within a kind of chemical synapse, there are 4 steps for neurotransmission, or the transmission of neurotransmitters into the synaptic cleft for reception on the postsynaptic cell. The first step is synthesis and storage. So some neurotransmitters are synthesized in the cell body and transported to the axonal terminal, and others are synthesized right in the axon terminal from raw materials that came from the cell body. The second step is the release of neurotransmitters. When an action potential reaches the axon terminal, it triggers voltage-sensitive calcium channels to open. Calcium ions flow into the presynaptic terminal, and an influx of calcium ions causes the vesicles to fuse to the presynaptic membrane. And the vesicles, once they fused with the membrane, they kind of push out their contents into the synaptic cleft, so now there are neurotransmitters in the synaptic cleft. And this is just an image from an electron microscope. You can see where the little arrows are pointing that vesicles have fused with the cell membrane and are pushing their neurotransmitters into this gap between the cells, the synaptic cleft. In the third step of neurotransmission, the postsynaptic receptor site is activated, so the neurotransmitters diffuse through this synaptic cleft, this gap, and bind to the receptors on the postsynaptic cell. Receptors are specialized protein molecules that are embedded in this membrane on this postsynaptic cell. And the postsynaptic cell responds in one of 3 ways. There can be an excitatory response, there can be an inhibitory response, and there can also be kind of a setting off another chain of chemical responses that are ultimately excitatory or inhibitory. So if you remember, in the nervous system, there are kind of like 2 main types of responses or ways of transmitting signals, and that's excitatory and inhibitory. So ultimately, all actions between neurons are excitatory or inhibitory. Okay, and this is just another image of the activation of receptor sites. The neurotransmitter is released into the synaptic cleft. It binds to the receptor. It's kind of like a key in a keyhole. The neurotransmitter doesn't actually go into this postsynaptic cell. It just binds to this receptor, which creates a response. So the fourth step of neurotransmission is that the neurotransmitters are deactivated. So after the neurotransmitter has bound to its receptor, one of four things can happen. Either the neurotransmitter diffuses away from the synaptic cleft, it just drifts away. It can degrade in the synaptic cleft, so enzymes or other chemicals can break down that neurotransmitter and make it inactive. Another common one is free uptake, so the neurotransmitter is transported back into the presynaptic cell for reuse later. And then astrocytes, which, if you remember, are glial cells, take the neurotransmitter and typically store it for later use. There are a number of types of synapses. For simplicity, I don't expect you to know all of these. For the purpose of this course, we're mostly going to be talking about axodendritic and axosomatic synapses. So axodendritic are the ones where an axon synapses with a dendritic spine or any part of the dendrite of another cell, and axosomatic is when an axon of one cell synapses with the soma or the cell body of another cell. Excitatory synapses are typically axodendritic, so typically when an axon is synapsing with a dendrite, that's more likely to be excitatory, whereas inhibitory synapses are typically on the cell body. There are four main criteria for determining if a chemical is a neurotransmitter. One, it has to be synthesized within the neuron. So neurotransmitters are always created or synthesized within the actual neuron. Two, when it's active, it must be released and create a response in the target cell. Three, the same response must be created every time, so it has to be consistent in the response it creates. And four, there has to be a way of removing the neurotransmitter from the action site after the chemical's work is finished, so reuptake or diffusion, etc. Notably, so these are kind of the classical criteria for determining if a chemical is a neurotransmitter. These days, researchers use the term neurotransmitter much more broadly, so you might, if you get further in your psychology career and see neurotransmitter being used more broadly, that's kind of a trend lately in neuroscience. Okay, so we're going to talk about the four classes of neurotransmitters now. These four classes are based on chemical composition. Different books classify neurotransmitters in different ways, so don't be thrown off if you see neurotransmitters being classified as 3 classes or 2 classes, etc. But for the purpose of this class, because it's how your book chooses to classify them, we're going to talk about small molecule neurotransmitters, peptides, lipid transmitters, and gaseous transmitters. Small molecule transmitters are kind of the classic neurotransmitters. Those are the ones you've probably heard about the most. And it includes most of these best known and most studied neurotransmitters. They're quick acting because they're small, and the main components to synthesize these neurotransmitters are derived from dietary nutrients. So this includes things like dopamine, serotonin, norepinephrine, which is also called noradrenaline, glutamate, GABA, histamine, etc. Quick note, your book goes into a lot of detail about the synthesis of small molecule neurotransmitters. For the sake of simplicity and time in this course, don't worry about the details of synthesis. Just know, in general, small molecule neurotransmitters are derived from dietary nutrients. And this is just a chart with a little bit more information about many of the most common small molecule transmitters. Okay, peptide transmitters, also called neuropeptides, can act either as neurotransmitters or hormones, depending on what part of the body they're acting in, the central nervous system or the rest of the body. As hormones, they respond to stress, they facilitate mother-infant bonding, they regulate eating and drinking, regulate pleasure and pain, and can also contribute to learning. They indirectly influence cell structure and function through synaptic receptors in the CNS, and they typically can't be taken orally as drugs. So some of the small molecule transmitters can be taken orally, so like dopamine, etc., can be taken orally as drugs, but these cannot because they're easily digested within the digestive system. Peptide neurons, you might be familiar with a number of these, so opioids are peptide transmitters, insulin, and then you might be familiar with oxytocin, which we're going to talk about on the next page. So oxytocin is a neuropeptide that facilitates social bonding. It also plays a role in childbirth and bonding of the mother and child, especially right after birth. And researchers have been looking at oxytocin as a potential medication for psychological disorders in which people are not as prone to social bonding or have social cognition deficits. So one of the more popular diagnoses that this is used for is autism. So oxytocin in the form of nasal spray, because again, you can't ingest it without digesting it too quickly. So children with autism in these studies are given this nasal spray, and it has been shown in a number of studies, actually, randomized controlled trials, that social interaction is improved in children with autism through the regular administration of this oxytocin nasal spray. And I'm not recommending this, but you can actually buy it on Amazon. But I'm not, again, not recommending this necessarily. Okay, so moving on, lipid transmitters. Lipids are a group of molecules that include fats, waxes, some steroids, vitamins, some vitamins. The most popular or most common lipid transmitters that you're going to hear about are endocannabinoids, which means endogenous cannabinoids. These endocannabinoids affect appetite, pain, sleep, mood, memory, anxiety, stress, everything you might associate with cannabis. Endocannabinoids can inhibit the release of both glutamate, which is excitatory, and GABA, which is inhibitory, and we'll get more into that later. Phytocannabinoids, that's like the active ingredients in marijuana, bind to the same CD1 receptors as endocannabinoids, so that's why you get a lot of the same effects in phytocannabinoids, which come from the outside, and endocannabinoids. Two prominent components of phytocannabinoids or cannabis are cannabidiol, which is usually called CBD, and then THC, which actually have potential medical benefits. So CBD, there's some evidence for treating treatment-refractory epilepsy, or epilepsy that doesn't respond to other epileptic medications. And there's actually some evidence, kind of preliminary trials, that indicate that CBD may have antipsychotic effects, so may be effective in treating psychosis. THC has been shown in some clinical trials to help with MS, pain that is mediated by the central nervous system, bladder dysfunction, and a number of other disorders. Notably, there's something called synthetic cannabinoids that are sold as drugs. It's also often called spice, and these are very bad. They are not like endocannabinoids. It's not the same thing as cannabis. It's not the same thing as endocannabinoids. It's usually an unpredictable combination of chemicals, very cheaply and inconsistently created. Side effects can be very serious, including seizure, prolonged psychotic episode, panic attacks, and vomiting. And the effects of the drug, or the positive effects of the drug, are not the same as cannabis. So synthetic cannabinoids is almost a bit of a misnomer and should not be associated with some of the more positive effects of cannabinoids. Gaseous transmitters include nitric oxide, carbon monoxide, and hydrogen sulfide. Nitric oxide and carbon monoxide activate metabolism, whereas hydrogen sulfide slows down metabolism. Okay, so now we're going to talk about receptors. There are two main classes of receptors. Again, receptors are these specialized proteins that are embedded in the cell membrane of the postsynaptic cell. The first class is ionotropic receptor. This acts as a direct binding site for neurotransmitters. It's a pore that regulates ion flow to change the membrane voltage quickly and directly. So a neurotransmitter binds to the ionotropic receptor, and then that receptor also acts as a pore that lets these ions in. And these are usually excitatory, which, if you remember from last chapter, means that they are likely to contribute to the triggering of an action potential. The second receptor type is metabotropic receptors, And this also has a neurotransmitter binding site, but it's not its own pore. So when a neurotransmitter binds to a metabotropic receptor, this the receptor indirectly causes cellular changes. So it might set off a cascade of chemical responses that opens a nearby pore or activates other chemical processes. And this is just another image of the ionotropic receptor, where you can see this red ball is the receptor. It binds to the receptor site, and this opens up the pore, allowing ions to enter. And conversely, in the metabotropic receptor, this transmitter binds to the receptor site, which causes what's called a G-coupled protein, but don't worry about that. So it creates chemical responses that in turn open up a pore, possibly a nearby pore, or it can create a series of chemical cascades which also open up a pore. But the end effect of both types of receptors are the same, that ions are allowed into the cell one way or another. There are different subtypes for each of these receptors. Don't worry about knowing all the different subtypes, just know that there are many different subtypes for both ionotropic and metabotropic receptors, and each subtype has slightly different properties. Receptors are also specific to different neurotransmitters, so there's like serotonin receptors, and there's a number of different types of serotonin receptors, etc. It used to be thought that any one neuron transmitted only one type of neurotransmitter. It's now known that a single neuron can use different neurotransmitters at different synapses. In terms of neurotransmission in the somatic nervous system, motor neurons use mostly acetylcholine as their neurotransmitter. They're called cholinergic neurons. In skeletal muscles, cholinergic transmission creates muscle contraction. And one of the main cholinergic neuron receptors is the nicotinic acetylcholine receptor, and it's named this because nicotine is close enough in structure to acetylcholine that it can also activate this receptor. So nicotine and acetylcholine can both activate the nicotinic acetylcholine receptor. Within the ANS, the autonomic nervous system, both the sympathetic and parasympathetic subdivisions of the nervous system are controlled by acetylcholine neurons coming from the central nervous system at different parts of the spine, as you can see in the figure to the right. In the sympathetic nervous system, which you might remember is the fight or flight part of the nervous system, acetylcholine neurons synapse with norepinephrine neurons, whereas in the parasympathetic, which is the rest and digest part of the nervous system, acetylcholine neurons synapse with other acetylcholine neurons. The enteric nervous system acts with autonomy, so the ENS can act without input from the CNS. It uses all four classes of the neurotransmitter, but serotonin and dopamine are particularly prevalent. So there are four main neurotransmitter systems that we're going to talk about in the CNS, and these are activating neurotransmitter systems. It's we'll talk about the cholinergic system, the dopaminergic, noradrenergic, and the serotonergic systems. These are comparable to a power supply in the brain, meaning these are sort of the main mechanisms of excitation in the brain. Neurotransmitters throughout the brain create different functions depending on where they are in the brain. So for example, dopamine in one area of the brain will create a different function than dopamine in another area of the brain. All of the systems we're going to talk about have widespread functions in the brain. So the first, the cholinergic system, is driven by acetylcholine. So acetylcholine is especially dense in the basal ganglia, which, if you remember from previous chapters, is associated with voluntary motor activity, habitual activity, procedural learning, and emotion. It also has a role, the cholinergic system has a role in attention, wakefulness, and memory. And a loss of neurons, especially in the basal ganglia, is associated with progressive dementia like Alzheimer's disease. So dopamine is the main actor in the dopaminergic system, and there are two main pathways. There's the nigrostriatal pathway, which is this pathway in purple at the picture on the upper right here, going through the frontal lobe and also the brainstem. And this maintains motor behavior. If there's too little dopamine flowing through this system, this pathway, then motor abnormalities are the result. So one disease that we talked about previously was Parkinson's disease. And in Parkinson's disease, there is not enough, there's an atrophy or a decrease in the amount of dopamine neurons in the substantia nigra. So if you look at this picture down here, you can see that in the normal individual, there are these dark grooves, these dark spots, and that's dopamine neurons. And in the individual with Parkinson's disease, there's a decrease. There are fewer of these dark spots, meaning there's less dopamine. There are fewer dopamine neurons in that area. The second pathway is the mesolimbic dopamine pathway, and this is the reward system of the brain, so associated, kind of like the pleasure center, people call it as well. And this is implicated in a number of psychological disorders, including addiction. This is also the pathway that's believed to be responsible for the runner's high. So if any of you are runners or do a lot of athletic activities, intense athletic activities, sometimes people experience this feeling of like a high, like almost you're on drugs when you're at a certain point in intense exercise activity, and this is actually the mesolimbic dopamine pathway being activated, so the reward center of the brain. And it literally is a response similar to taking drugs. So the name runner's high is actually a pretty appropriate term. Okay, and if you look over on the right, the bottom right picture here, you can see that the reward center of the brain, this mesolimbic pathway, is dysfunctional in a number of medical and psychological disorders. So in the normal brain, you see these red spots, which means high levels of dopamine transmission. The yellow means, I think, like moderate, and the green means low levels of dopamine transmission. So a normal amount, you have these red areas of high activity. In the brain of an obese person, a very obese person, there's some indication that people who are very obese have less of a pleasure response, so less activity in the reward system when they eat food. The dopamine neurons, there isn't as much dopamine activity in the brain of an obese person eating food in general compared to somebody who is not obese. And then we have the brain of a cocaine user. Cocaine decreases the regular transmission of dopamine in the brain, and similar, somebody with alcoholism has dysfunctional reward system in their brain. The dopaminergic system has long been implicated in schizophrenia, which is this psychological disorder characterized by hallucinations, or seeing things and hearing things that aren't there, as well as delusions, believing things that are not true. There are other neurotransmitter systems involved, but dopamine was one of the first that we discovered, and it's definitely plays a big part in schizophrenia. In schizophrenia, there's excessive dopamine in the mesolimbic system, which causes some of these positive symptoms like hallucinations and delusions, and not enough dopamine in other areas of the brain, which cause what we call the negative symptoms, which is lack of motivation, inability to experience pleasure, etc. Antipsychotic drugs decrease the overall amount of dopamine transmission in the brain. So antipsychotic drugs are meant to normalize dopamine or decrease the amount of transmission to avoid these positive symptoms of hallucinations and delusions. And they don't do much for negative symptoms because negative symptoms are already caused by not enough dopamine in the brain. Sometimes antipsychotic drugs can have motor side effects. So as we talked before, dopamine transmission has a big part to play in motor activity. So reducing dopamine transmission can have the side effect of causing motor abnormalities in people with schizophrenia. So we see side effects called Parkinsonian symptoms, in which people on antipsychotic drugs will actually have the motor abnormalities similar to Parkinson's disease. In the noradrenergic system, this is the norepinephrine system, So norepinephrine, which is also called noradrenaline, is very closely related to epinephrine or adrenaline, which you might be familiar with. And in the noradrenergic system, neurons are stimulated to change structure and create learning. Too much norepinephrine is one possible cause of mania and bipolar disorder, and too little norepinephrine is one cause of depression or ADHD. So people who have bipolar disorder typically go through these states of mania or really, really high activity and feelings of like euphoria or intense pleasure, and then drop down into states of depression or intense sadness and inability to experience pleasure. You can see on the bottom right here, this is just general brain activity of one individual who is in a depressed state, and you can see from the green and the yellow that there were lower levels of activity in the brain during the depressed state as compared to this manic state where there's high levels of activity with the red and more yellow. And then back to the depressed state, it looks like just a week or so later with lower activity. So the serotonergic system is, of course, serotonin flow through the brain. Serotonin is often abbreviated as 5-HT. Serotonin plays a role in wakefulness. So more serotonin, when you start to feel sleepy, this is usually increased levels of serotonin in your brain. Serotonin also plays an important role in depression. There may be two subtypes of depression, one related to serotonin, the other related to norepinephrine. And consistent with this, there are two main types of antidepressants. The most common are called SSRIs, or selective serotonin reuptake inhibitors. And then there's also this slightly newer and less common kind called serotonin norepinephrine reuptake inhibitors. Decreased serotonin is also related to obsessive compulsive disorder, which is a psychological disorder in which a person compulsively repeats rituals such as washing their hands and has obsessive thoughts, such as obsessively thinking about whether they locked the door. So there are a few more neurotransmitters that I wanted to talk about that your book didn't go into too much detail on. Two of them are glutamate and GABA. Glutamate and GABA are very closely related to each other. They're kind of called the workhorses of the brain. These are two neurotransmitters that act in opposition to each other. So glutamate is the main excitatory neurotransmitter in the brain. It plays an important role in learning and memory and synaptic plasticity. Too much can cause seizures, and too little can cause a coma. GABA is the main inhibitory neurotransmitter in the brain. Medications that increase GABA have anxiety-reducing effects, can be relaxing, and also anticonvulsive. So when somebody has epilepsy, they might take a medication that increases GABA in the brain. Side effects of drugs that increase GABA are inhibition of memory formation. So you can see how glutamate, increased levels of glutamate have an important role in memory, and then GABA kind of acts in opposition to this, where increased GABA might inhibit memory formation. We're going to talk more about drugs that target these two in the next chapter. And then there's histamines, which you might be familiar with, antihistamine drugs that are used for allergies. So histamine is a neurotransmitter that mediates arousal and wakefulness. It can cause constriction of the smooth muscles. When somebody's having an allergic reaction, there's increased histamine transmission, and the esophagus can constrict, causing breathing issues. And antihistamine drugs prohibit histamine transmission, so they reduce this constriction of the esophagus, for example. And as histamine is associated with wakefulness, antihistamine drugs are associated with sleepiness. So overall, neurotransmitter systems are related to each other and they act together. A change in one neurotransmitter system can, and often does, cause a change in another one. Many psychological disorders involve the dysregulation of multiple systems. So many people will say like schizophrenia is caused by dysregulation in dopamine, and that's true, it's part of the puzzle, but it's not the whole picture. For example, anxiety has many neurotransmitters that have been implicated, such as serotonin, dopamine, norepinephrine. Depression and mania are both associated with serotonin and norepinephrine. The interaction between neurotransmitter systems is complex and often unclear. It's more difficult to study interactions between these systems. And these sort of overlapping functions and interactions between neurotransmitter systems may be one reason for the prevalence of what we call comorbidity in psychological disorder. So often somebody with one psychological disorder will have another one. So there are disorders which are often seen paired together. So examples of this would be someone with depression is at a greater risk for anxiety. People with schizophrenia are at greater risk for anxiety and depression. Somebody with OCD is more likely to have depression, etc. Okay, so moving on to the adaptive role of synapses and learning and memory. Larger brain, such as the human brain, means more connections between neurons, means a greater potential for plasticity. So more connections means more possibility of changes, and this means that larger brains lead to more adaptability in that organism. Learning we will operationally define as a relatively permanent change in behavior that results from experience, and learning, as we talk about it in neuroscience, is mediated by structural changes in synapses. Research on the neurobiology of learning is often done with this animal called aplasia, which is also called sea slugs or sea bunnies. And these are some pictures of some sea slugs I found when I was living briefly in Australia. It was a neuroscience grad student's dream, I think, to go into the ocean and find aplasia. They're actually pretty cute, and you can see on top these gills, which are referenced a lot in your book in terms of these learning experiments that have been done with aplasia. So don't worry too much about the nitty-gritty details about research with aplasia. What you mostly need to know is two main mechanisms of learning. Habituation is the first one, which is an unconscious learned behavior in which the response to a stimulus weakens with increased or repeated presentations. So, for example, you might notice the sound of a fan when you walk into a room, but over time you become habituated to the sound and you no longer notice it. And what's happening in your brain, your neurons are actually not responding as much to that sound as they were when you first walked into the room and noticed it. So, in aplasia, this is tested with putting like jets of water, which is kind of an adverse stimulus, on the gills of the top of this little sea bunny. And the aplasia, typically when you squirt water at it, it'll withdraw from the water jet, like close its gills. But over time, the aplasia, if you repeat this stimulation of the water jet, the aplasia stops doing this, stops retreating its gills. So what's happening neurobiologically, there's typically an influx of calcium ions in response to the action potential, and this influx of calcium ions actually decreases in the aplasia cells. The motor neuron receives less neurotransmitter from the sensory neuron, and the excitatory postsynaptic potentials in the motor neuron is smaller, meaning it's less likely to create another action potential. Sensitization is kind of the converse of habituation. This is an unconscious learned behavior in which response to a stimulus strengthens with repeated presentations. For example, if you're talking to someone and they say like every fourth word, you might find yourself becoming increasingly annoyed to the word like. And if we looked at this neurobiologically, you would actually see increased response in your neurons to the word like, the more it annoys you. So what's happening is interneurons, in the case of the aplasia, it's getting this water jet, but it's also getting a shock to its tail, which it finds very averse. And so the interneuron receiving the shocked input from sensory neuron releases serotonin onto the sensory neuron axon. The serotonin prolongs the effect of the action potential on the sensory neuron, which leads to an increased neurotransmitter release and greater depolarization of the postsynaptic membrane. And this creates behaviorally an increased response to the stimulus. Learning can also be conceptualized as a change in the number of synapses. So for a permanent change in behavior, structural changes have to happen. So neural changes have to be long-lasting. If you study for just one night, you don't learn as well as studying for the same amount of time over the course of a week, because if you study for a long amount of time, structural changes happen. Synapse numbers decrease in the habituated sea bunnies and increase in the sensitized sea bunnies, for example. There's a second messenger called cAMP that plays an important role in signaling DNA to create structural changes associated with learning. Disruption of the secondary messenger leads to interruptions in learning, and disruption may be involved in some prefrontal disorders where we see problems with learning, including age-related learning disorders, so disorders where people are less able to remember and learn things as they get older, and also ADHD, attention deficit hyperactivity disorder.